Hello and welcome all of you to my channel Omni Bioentrance, and today we will be discussing University of Hyderabad MSc Entrance Examination question paper for the subject Plant Biology and Biotechnology. I will be discussing only the biology part. Some chemistry questions have been asked which I have not attempted. Then, which of the following statements about the nature of enzyme catalysis is correct? An enzyme can change the equilibrium position of the reaction it catalyzes by lowering the energy of activation of that reaction. An enzyme cannot change the equilibrium position of a reaction. An enzyme can lower the energy of activation of the reaction it catalyzes by increasing the molecular collision between molecules. So that is also not correct. An enzyme lowers the free energy difference between substrate and products, but it cannot change the equilibrium position of the reaction it catalyzes. That is also wrong. An enzyme cannot change the equilibrium position of the reaction it catalyzes, but it lowers the energy of activation of that reaction. Answer D is correct. Then, which of the following is not a reducing sugar? Now, sucrose, because of no free aldehyde or ketone functional group. So sucrose is not a reducing sugar. Then, which of the following is most likely to accelerate the evolution of proteins with new functions? So exon shuffling. Exon shuffling is a molecular mechanism for the formation of new genes. It is a process through which two or more exons from different genes can be brought together ectopically, or the same exon can be duplicated to create a new exon intron structure. There are different mechanisms through which exon shuffling occurs, that is transposon mediated exon shuffling, crossover during sexual recombinations of parental genomes, and illegitimate recombination. So in exon shuffling, exons can recombine to create novel proteins. So which of the following is most likely to accelerate the evolution of proteins with new function is through exon shuffling? Answer is option A. So coming to the next question, to make 1 ml of 20 millimolar ATP, how much of 10 millimolar ATP stock should be taken? So they have given 20 millimolar ATP as the stock, as the working concentration, and for preparing that, how much of 10 millimolar ATP stock should be taken? So 10 millimolar into X is equal to 20 into 1, so X is equal to 2 ml. They have given the value in microliters, units in microliters, but you will get it in ml only. So 2 microliter, it might be a printing mistake, it might be 2 ml, it might be there, but answer is in 2 ml. If it is in microliter, it has to be 2000 microliter. So the answer is 2 ml. So coming to the next question, which of the following single strands would be part of a palindrome in double-stranded DNA? So a palindromic sequence is a nucleotide sequence in the double-stranded DNA whereby reading in the 5' 3' direction in one strand is identical, has got an identical sequence in the same direction in the complementary strand, that is 5' to 3'. So both the strands from 5' to 3' will have identical sequence. That is called as a palindrome. So here we have got ATG, ATG. Now the complementary strand, that is TAC, TAC, it will not have a palindrome. Similarly CTAA, TC, GATT, AG. So that is not a palindrome. It is not at all equal to this particular strand in 5', 3' direction. Similarly this one also, but if you take GAA, TTC, if you read it in this direction, that is the complementary strand from 5 prime to 3 prime direction, that is GAATTC. So that is a palindrome. So the answer here is option A, that is GAATTC. Arrange the following processes in cell cycle in correct order. So they have given the different phases of the cell cycle. We have to arrange it in the correct order. So the answer is, you have got first the prophase, then comes the metaphase, then comes anaphase, then comes telophase, and finally cytokinesis. So answer is option B. Then which one of the following is not a sulfur containing amino acid? So proline is not a sulfur containing amino acid, whereas methionine, cysteine, homocysteine all are sulfur containing amino acids. Then the net gain of energy molecules from glycolysis pathway of one glucose could be. So it is 2 NADH molecules and 2 ATP molecules. So answer is option D. The next one. Which of the following statement is incorrect for bryophytes? They have a dominant gametophyte generation. Fertilization occurs in water. Presence of non-lignified vascular system. Occurrence of gametophyte independent sporophyte. So that is the incorrect statement of bryophytes. It is independent gametophyte sporophyte. So answer is option D. Then how many mitotic cell divisions are required to produce 2048 cells from a single cell? So 11 mitotic divisions are required because 2 raised to n is equal to 2048. So 2048 is nothing but 2 raised to 11. So 2 raised to 11, so the number of mitotic divisions required will be option B, that is 11. Which type of bonding is responsible for the secondary structure of proteins? So for secondary structure you need hydrogen bonding between CONH group of peptide bonds. That is in the case of alpha helix, beta pleated sheets and all. Then next one, join the identify the mismatch. So vessels, Welwitschia, Menas, Tiger Reserve, Sacred Groves, ex situ conservation. Sacred Groves is not ex situ conservation, it is in situ conservation. So that is the mismatch. And Ramar site is Renuka Lake. Okay. Then match the names or features present in list A with their family from list B. So Pollinia, it is found in Asclepidaceae family. So 1D. Then neem. Neem is Miliaceae. Answer is option C. 2C. Then nightshade. Nightshade is 3A, that is Solanaceae, and parallel venation is Poaceae. So the answer is option A. 1D, 2C, 3A, 4B. Then next one. Which of the following is incorrect in students' t-test? It compares two means of two groups to find out the significant difference between them. It indicates that if the significant difference could have happened by chance, a large t-score is indicative of higher similarity between the groups. So the answer is, this is incorrect. A large t-score is indicative of higher similarity between the group, because higher values of t-score indicate that a large difference exists between the two sample sets. Smaller the t-value, the more similarity exists between the two sample tests. Then low p-values in a t-test are good because they indicate you data did not occur by chance. Okay, so the answer is option C. Then chromosomal crossing over during meiosis occurs during. So chromosomal crossing over occurs during the prophase 1 of meiosis. Answer is option A. Which of the following is not a form of asexual reproduction? So syngamy. Syngamy, it is sexual reproduction by union of gametes. So answer is option D. All the others are asexual. Then match the following Nobel laureates given in the list A with their discoveries present in list B. Then Yoshinoro Ohusumi. So that is autophagy. Frederick Sanger is for determination of the base sequences in nucleic acid. So 2C, 1D, 2C. And Peter Michel is for chemiosmotic theory, and Marshall W. Nierenberg is interpretation of genetic code. So 1D, 2C, 3A and 4B. So answer is option B. The next one, match the characters present in the list A with the correct plant names given in list B. Anomalous secondary growth. Anomalous secondary growth is seen Boyer-Havia. So 1D. Then epidermal fiber. Epidermal fiber is in the case of cotton. Phloem fiber in the case of linseed, and hemp fiber in the case of cannabis. So answer is option C, 1D, 2C, 3B and 4A. The next one, match the plant names present in the list A with the corresponding common name or feature in the list B. So Funaria is rhizoid, 1D. Then Equisetum is a xerophytic fern. Salvinia is aquatic fern, and Dicksonia is a tree fern. So answer is option B, that is 1D, 2C, 2B and 3B and 4A. Answer is option B. Next one, match the algae from list A with the products extracted from them presented in list B. So Dunaniella salina, it is beta carotene. 1C. Macrocystis pyrifera, iodine and potassium. 2A. Glaricellaria bursa pastoris, it is agar-agar. And Chondrus crispus is carrageenan. So answer is option D, 1C, 2A, 3D, 4B. Next one, the main role of phragmoplast during cytokinesis is. So phragmoplast in plant cell specific structure that forms during the late cytokinesis. This complex assembly of microtubules, actin filaments and associated molecules act as a framework for cell plate formation or cell plate assembly, and then subsequent formation of the future cell by separation of the two daughter cells. So answer is option C, supports cell wall. Answer is option C. Then phages that show lysogenic cycle are called. So temperate phages are some bacteriophages which display a lysogenic life cycle. So lysogeny, it is characterized by an integration of the bacteriophage nucleus into the bacterial genome, and it is called as the prophage at that stage. Then in this condition, the bacterium continues to live and reproduce normally. Okay, so that is the case in case of lysogenic life cycle. So answer is option C. Phages that show lysogenic cycle are called temperate phages, and bacteriophages that use only the lytic cycle, they are called as virulent phages. The next one, which of the following statements about the competitive inhibition of an enzyme catalyzed reaction is correct? So competitive inhibition occurs when molecules which are very similar to the substrate molecule bind to the active site and prevent the binding of the active site. So in this case, the Vmax of the reaction, it remains unchanged in the presence of a competitive inhibitor, whereas Kmax changes. So answer is option C, that is the Vmax of the reaction remains unchanged in the presence of a competitive inhibitor. The next one, in plants, major site for de novo biosynthesis of fatty acids is. So answer is option A, plastids. De novo fatty acid biosynthesis mainly take place in the plasticidal compartment from acetyl-CoA, which is a direct product of photosynthesis. So answer is option A. The next one, Which of the following act as a precursor for porphyrin formation? So porphyrin synthesis is the process that produces heme, and heme synthesis occurs partly in mitochondria and partly in the cytosol. Heme biosynthesis starts in the mitochondria with the condensation of succinyl-CoA from citric acid cycle and an amino acid that is glycine. So answer is option C, that is succinyl-CoA and glycine. So porphyrin precursors are succinyl-CoA and glycine. Then which of the following microorganisms leach metals out of rock ores and can accumulate silver? So they are Thiobacillus peroxidans. Answer is option B. Then a genetically distinct geographic variety within a species which is genotypically adapted to specific environmental condition is called. So it is called as ecotypes. Answer is option C. Then the ecological species concept. Ecological species is a concept of species in which species is a set of organisms adapted to a particular set of resources called a niche in the environment. Then ecophenes are the variety of phenotypes from a single genotype that can be observed in a population within a particular habitat. And subspecies, it refers to one or more population of species living in different subdivisions of the species range and varying from one another by morphological characteristics. So that is called as subspecies. Next one. Which of the following statement about the mechanism of synthesis of fatty acid is correct? So acetyl-CoA is the active donor of 2 carbon atoms in fatty acid synthesis. So it is malonyl-CoA is the active donor of 2 carbon atom in fatty acid synthesis, not acetyl-CoA. So answer is option B. That statement is correct. Then fatty acid synthesis is the reverse of beta oxidation of fatty acid. That is also not correct. And coenzyme A is the acyl group carrier of intermediates in fatty acid synthesis. That is also not correct. Because the acyl group carrier of the intermediates in fatty acid synthesis is not coenzyme A but a protein called acyl carrier protein, to which the growing fatty acyl chain is attached. Then next one. Which of the following is not an example of a biofilm? So human microbiome. Biofilm, what is biofilm? They are collective of one or more types of microorganisms that can grow on many different surfaces. So human microbiome is not an example of biofilm. Then next question. The apparatus used for measuring rate of transpiration is called. It is called as photometer. Answer is option B. Then if the occurrence of one event means that another cannot happen, then the events are then mutually exclusive. We are told that if one event occurs, then another cannot happen. When the occurrence of 2 events are not possible simultaneously, then the events are called mutually exclusive event. So if the occurrence of one event means the other cannot happen, the events are mutually exclusive. Answer is option C. The next one. Resistant genes found in the commonly used cloning vector PBR322. They are tetracycline and ampicillin. If you see the structure of PBR322, you can see this resistance gene in the map. Then Striga is now Striga or witchweed, it is a partial root parasite of sugarcane, jowar, maize, cereal and millets. So Striga is a partial root parasite. Answer is option B. Then if the respiration rate is higher than the rate of photosynthesis, the plant will. If the respiration rate exceeds the rate of photosynthesis, the plant will be using more oxygen than it can produce. Thus the plant will be starved of food supply and finally die of starvation. So the answer is option A, that is die of starvation. Then synaptonemal complex during meiosis is formed in the substage zygotine. Answer is option C. So coming to the next question. In the morning, the fresh weight of plant is usually greater than that in the evening because. So in bright sunlight, that is during the morning, the stomata remain fully open and transpiration takes place through stomatal pores. At night, stomata remains closed, so transpiration is reduced. So answer is plants transpire less in the night. Answer is option D. Then George Beadle and Edward Tatum received a Nobel Prize in 1958 for demonstrating the interaction of genes in biochemical pathways in the haploid fungus Neurospora. In their study, they isolated mutants that were affected in the synthesis of. So, in their experiment, Beadle and Tatum, they isolated mutants that were affected in the synthesis of arginine. Then next one. Drosophila virilis is a diploid organism with 6 pair of chromosomes, 12 chromosomes altogether. The number of chromatids and the chromosomes that are present in metaphase 2 of meiosis. So the metaphase 2 take place in the meiotic division will have only half chromosomal number in their cell as their number has already been turned down in the first meiotic phase. Therefore, the number of chromosomes in this phase will be 6 and the chromatids will be 12. So the answer is option B, 12 chromatids and 6 chromosomes. The enzyme responsible for initiating DNA replication in prokaryotes is. So initiation of replication process is done by the primase. The primase synthesizes RNA primer for DNA polymerase 3 so it can begin the replication process. So answer will be option D, that is primase. Then next one. An individual of genotype, so they have given A, B, C, D, E, F is test crossed. Assuming that the loci undergo independent assortment, what fraction of the progeny are expected to have the genotype? So they have given the genotype of the expected progeny. Now these are the offsprings. It is test crossed means it is crossed with its recessive parent. So all the genes will be small letters, will be written in small letters. So if you now what we need here is we need capital A small a, that is the first genotype we have to see for. So if you cross this, that is the first parent is having both capital A with the recessive parent, that is in the case of test cross. So small a, we will see that capital A into capital A, it will give capital A small a, that is only one type of offspring you will get with this particular combination. Then if you cross, you get only one offspring. Then similarly if you cross capital B with small b, you will get only one genotype, and if you cross capital C with small c, you will get two genotypes. Similarly, in the case of capital D, small d, you will get only one phenotype, and capital E, small e, you will get two, and capital F, small f also you will get two. So, assuming that the loci undergo independent assortment, what fraction of the progeny are expected to have? So, in all these cases, only one progeny will be available, that is capital A, capital B, small b. But here, out of the two progenies, only capital C, will be shown by half of the progeny. Similarly, in the case of this E crossing also, only half of the progeny will be showing capital E, small e, and here in the case of F, half of the progeny will be showing capital F, small f. So multiply everything, 1 into 1 into half into 1 into half into half, that will give you 1 by 8. So the answer here is 1 by 8. So what fraction of the progeny are expected to be of this genotype is 1 by 8 of the progeny will be of this particular genotype. Then the question is repeated, so I repeated it while copying it, that's why it has come. I have already explained that, just in the last slide. So it is 12 chromatids and 6 chromosome. Okay. And the same here also. This also I repeated by mistake. It is primase the answer. Then gibberellins were discovered during scientific studies of foolish seedling disease caused by the fungus in the following plant species. So gibberellins were first isolated from the fungus Gibberella fugicori, the cause of the foolish seedling disease of rice. So answer here is Oryza sativa. Answer is option C. Then one of the primary reasons for the loss of biological activity in aquatic and marine dead zone is, so dead zones occur because of a process called eutrophication, which happens when a body of water gets too many nutrients such as phosphorus and nitrogen. So eutrophication, is an increase in chemical nutrients in the water, leading to excessive blooms of algae that deplete underwater oxygen. So the one of the reason for the loss of biological activity in marine and dead zone is depleted levels of oxygen due to overgrowth or blooms of algae. Answer is option A. So coming to the next question, which of the following statement is incorrect about passive transport? It involved movement of ions across cell membrane. The rate of passive transport depends on the permeability of the cell membrane. It does not require cellular energy for transportation. Then it moves solute from area of low concentration to area of high concentration. So that is the incorrect statement, because passive transport moves solutes from an area of higher concentration to an area of lower concentration. Then two linked genes are separated by a distance that is exactly 10% of the cells undergoing meiosis have one crossover between the genes and 90% have no crossover. The percent recombination between the genes, so it is 10%. Answer is option B. Then Queen Victoria was a carrier of hemophilia, a sex-linked disease. Which of the following statements is true? Hemophilia would have occurred more in her males than female descendants. So that itself is correct. Queen's father must have had hemophilia. All of her sons would have had hemophilia. All of her daughters would have had hemophilia. So answer would be option A. Then on immersing an iron nail into CuSO4 solution for few minutes, you will observe. So the color of the solution changes to green. This is because of its high reactivity. The iron ions slowly displaces the copper ions that form in the solution. So iron ions undergo a mixed solution while the copper ions are deposited on iron nails due to the displacement reaction. So CuSO4 plus Fe will give you FeSO4 plus Cu. So the color of the solution changes to green. Answer is option D. Which of the following statement is true? So proteins are synthesized away from carboxy terminus. No, it is always from the amino terminus to carboxy terminus. So answer is option B. So that is the statement that is true. Then which of the following RNAs are involved in splicing of introns from primary genomic transcripts? So ribosomes have rRNA and proteins. So which of the following RNAs are involved in splicing of introns from primary genomic transcripts? So rRNA. Answer is option A, that is ribosomal RNA. Then next one, endosperms are certain bacterial spores that enable them to survive in adverse conditions. Wait a moment, I made a mistake here. Okay, which of the following are involved in splicing of introns from primary genomic transcripts? So the answer is it is small nuclear RNA or snRNA that is involved, because splicing is catalyzed by the spliceosome, a large RNA protein complex composed of 5 small nuclear riboproteins or snRNPs. Assembly and activity of the spliceosome occur during transcription of the pre-mRNA. The RNA components of snRNPs interact with the intron and are involved in catalysis. snRNA and snRNP differ in that snRNAs are small nuclear RNA molecules, while snRNPs or small nuclear ribonucleoproteins are small nuclear RNA molecules with protein. So the answer is option D, small nuclear RNA. I made a mistake there. Then endospores are bacterial spores that enable them to survive in adverse conditions. Answer is option A. Then which of the following does not contain nucleic acid? So nucleic acid is not present in case of ribosomes. So answer is option C. Ribosomes have rRNA and proteins. So sorry, it is not ribosome, it is endoplasmic reticulum that does not contain nucleic acids. Option D, because ribosome has got rRNA. rRNA is a nucleic acid. So it has nucleic acid as well as protein. So endoplasmic reticulum does not contain nucleic acid. Then match the names given in list A with the class of major chemical compound present in list B. So natural rubber is terpenes. Okay, major chemical compound present is terpene. Then black pepper, it is piperine. Golden rice, it is beta carotene, and Papaver somniferum, it is morphine. So answer is option C, that is 1C, 2D, 3B and 4A. Then during secondary growth, the cells of the cortex of a dicot plant turn meristematic, giving rise to a cork cambium known as, it is called as phellogen. Answer is option C. Then Hydra belongs to the phylum Cnidaria. Answer is option A. Then promoter is, it is an upstream DNA sequence of a gene which is recognized by RNA polymerase in order to initiate transcription. So answer is option B. A technique that separates and resolves molecules according to their net charge in an electric field, usually on solid or semi-solid agarose medium, is called. It is called electrophoresis. Answer is option B. Then next one. A pigment containing sensory protein found in specialized light receptor cells called rhodstats is required for vision in dim light is. So it is rhodopsin. Answer is option C. Seed dormancy inhibited mechanically by causing seed coat injury is called. It is called as scarification. Answer is option A. Stratification, seeds are moistened and exposed to oxygen for variable period at low or high temperature. That is stratification. The answer here is scarification. Answer is option A. The next one. In which of the following phyla are the sperms non-motile? So in the phyla Neophyta. Answer is option C. The sperms are non-motile. Then the tDNA of one of the following bacterium is disarmed to use as a vector to clone gene sequences. So the tDNA of Agrobacterium tumefaciens is disarmed to be used as a vector to clone gene sequences. Answer is option. Next one. A multi-layered epidermis consisting of non-living compact cells with lignified strips of secondary wall to provide support, prevent water loss and assist in the plant in absorbing water is called as, velamen. It is seen usually in the case of epiphytes. Then, which stage of the plant development has no respiration? So, the germinating seeds. Because to fulfill the high energy needs of a germinating seedling, cellular response increases as a seed emerges from dormancy and begins germination. So the stage of plant development having more respiration is germinating seed. Answer is option A. Then next one, electron transport system happens in which part of mitochondria? So the inner membrane of mitochondria, the electron transport system take place in the cristae present in the inner membrane of mitochondria. So answer is option A. What is the role of kinase enzyme? So kinase is a kind of enzyme that transfers phosphate groups from high energy donor molecules, that is such as ATP, to specific target molecules, that is substrate, and the process is called phosphorylation. So kinase is addition of phosphate groups. Answer is option C. Then, what is the prosthetic group in chromoprotein? A chromoprotein is conjugated protein that contains a pigmented prosthetic group or cofactor. So, hemoglobin contains heme cofactor, which is the iron containing molecule that makes oxygenated blood appear red. So, what is the prosthetic group of chromoprotein? It is pigment. Answer is option D. Then next one. What is the byproduct in light induced photosynthesis process? It is ATP and NADPH. Answer is A. Mendel's tall dwarf alleles in P is an example of a single gene locus that can control the level of bioactive. So that can control the level of bioactive gibberellin. Answer is option C. Then phylogeny and interrelationship found between taxa on the basis of number, type and range of chromosomes is. It is called as cytotaxonomy. Answer is option A. Then next one, viroids have. So viroids are a group of pathogens of some higher plants that are smaller or simpler than any of the known viruses. They consist of single-stranded linear or circular RNA molecule without a protein coat. So the answer here is option C, that is single-stranded RNA not enclosed by protein coat. So a research scholar has newly joined a biology lab for his PhD. His supervisor explained the work and asked him to conduct an experiment with the given biomolecules and few chemicals or buffers as per given protocol. After mixing all the components, he suggested him to put the tubes at 95 degrees Celsius for 5 minutes, followed by 30 cycles of 95 degrees Celsius for 1 minute, 60 degrees Celsius for 30 seconds, 72 degrees Celsius for 1 minute, followed by 72 degrees Celsius for 4 minutes at 4 degrees Celsius for infinite time. Which experiment his supervisor suggested to perform? So here he was given certain biomolecules as well as certain buffers, and after mixing the components, he suggested them to put the tubes at 95 degrees for 5 minutes. That is when the denaturation of DNA takes place. Followed by 30 cycles of 95 degrees for 1 minute, denaturation of DNA. Then 60 degrees Celsius for 30 seconds, that is annealing. 72 degrees Celsius for elongation. So the experiment that the student is asked to perform is polymerase chain reaction. Answer is option C. Which of the following statement is false? So TMB has double-stranded RNA molecule. It is not double-stranded, it is single-stranded RNA molecule. So answer is option B. Then next one. Endosperm formation begins with. So the fertilization of polar nuclei, that is the fusion of secondary nucleus or the polar nucleus with the sperm give rise to primary endosperm nucleus, which is genetically triploid. So the endosperm formation begins with fertilization of the polar nuclei with the sperm nuclei. So answer is option D. Then next one. Which of the plant mitochondrial electron transport chain complex is not involved in pumping of H positive from mitochondrial matrix to inner mitochondrial membrane space? So if you go through the electron transport chain, you can see that succinate dehydrogenase complex is the one that is not involved in pumping of H positive ions or complex. It is also called as complex 2. Then next one, in angiosperm, the free nuclear division takes place during. So free nuclear division occurs in the primary endosperm nucleus that give rise to nutritive tissue or the endosperm. So the endosperm formation, answer is option B. Meiosis cell division is mainly responsible for. So it is mainly responsible for maintaining the number of chromosome constant from one generation to other. So answer is option D. Then next one, micronutrients are. So micronutrients are required by plants in smaller amounts. So answer is option B. The combination of pressure potential and solute potential is. So it is called as water potential. Answer is option A. So water potential, it is a measure of the ability of water molecules to move freely in solution. As more solute is added, water potential is decreased because the movement of water molecule is restricted by the solute particles. Then pressure potential also increases water potential. Thus water potential is a combination of solute potential and pressure potential. A common adaptation of aquatic plants is the formation of parenchyma. Answer is option C. Then the mature female gametophyte of an angiosperm is. So the female gametophyte is additionally usually called as the embryo sac or megagametophyte. The male gametophyte is called the pollen grain or microgametophyte. And it is formed inside the anther, and it comprises of two sperm cells encased inside a vegetative cell. So that is the female gametophyte and male gametophyte of an angiosperm. So it is the female, so they have asked the mature female gametophyte of an angiosperm is embryo sac with 8 nuclei and 7 cell. Now how these 8 nuclei and 7 cell came, I have already discussed in one of my previous question papers. Okay, you can. Then next one, which of the following is an essential element for all plants? So it is molybdenum, that is an essential element for all plants. Then in monocots, phloem is composed of specialized cells including. So phloem of monocots, they lack phloem parenchyma, so that we have to take into consideration. So they have asked for monocot, the phloem is composed of. So it is composed of sieve tubes, companion cells and phloem fibers. So answer is option B. Then next one, the softwood of conifers can be distinguished from the hardwood of angiosperms by the absence of. Then softwood, it mainly composed of tracheids and wood sap or parenchyma. Then wood fibers are absent and vessels are absent. So due to absence of vessel, softwood is also called non-porous wood. So softwood of conifers can be distinguished from hardwood of angiosperm by the absence of vessel elements. Answer is option A. Then the function of leg hemoglobin in the root nodules of legume is. So leg hemoglobin, just like hemoglobin, it is also a red pigment in the root nodules of leguminous plants such as soybean, and it's involved in nitrogen fixation. It acts as an oxygen scavenger. So answer is option D, that is oxygen scavenging. Then next one, backpack yak are generally used in molecular biology labs. These are, so they are different type of vectors. So BAC is bacterial artificial chromosome, is a DNA construct based on a functional fertility plasmid or F plasmid used for transforming and cloning in bacteria, usually E. coli. Then PAC is also produced from the DNA of P1 bacteriophage and it contains packaging site. So PAC phage artificial chromosome. PAC is phage artificial chromosome. Then YAC is yeast artificial chromosome, can be used to express proteins that require post-translational modification. So that is YAC. Then next one. In the evolution of land plants, sporophytes became dominant over gametophytes due primarily to what adaptation? So the evolution of seeds allowed plant to decrease their dependency upon water for reproduction. So seeds contain an embryo that can remain dormant until conditions are favorable and when it grows into a diploid sporophyte. So the evolution of land plants, sporophytes became dominant over gametophytes due to what adaptation? That is seed adaptation. So answer is option D. Then reindeer mosses. So reindeer moss is Cladonia rangiferina. Answer is option A. Then next one, which among the following has not been released as transgenic crop in the market so far? So tomato, longer shelf life, the transgenic is and is named flower saver. Then apple, the Arctic apple, it resists browning, and papaya virus resistance. So pepper has not been released as a transgenic crop in the market so far. The next one, vivipary is seed germination inside the fruit while still attached to the parent plant. Answer is option B. Then vernalization is the process whereby flowering is promoted by. So vernalization is the process whereby flowering is promoted by cold treatment given to a fully hydrated seed or a growing plant. So answer is option A, that is cold treatment given to a fully hydrated seed. Answer is option A. Then next one. Which of the following components is not essentially required for in vitro molecular cloning technique? So DNA, out of these DNA ligase plasmid restriction enzyme are required, whereas DNA topoisomerase is not required, as these are enzymes that participate in the overwinding or underwinding of DNA. Answer is option. Then next one. Match the type of cell wall material given in the list A with the group of organism it is present in the list B. Answer is option. Okay, we'll see. First one, chitin. Chitin is fungi. 1C. Then lignin. Lignin is angiosperms. 2D. Peptidoglycan is bacteria and alginic acid is brown algae. So the answer is option C, that is 1C, 2D, 3A, 4B. Then next one. What is the natural function of restriction enzyme? So it is protecting bacteria by cleaving the DNA of infecting virus. This is also a repeated question. Answer is option A. Then next one. UNFCCC stands for. So it stands for United Nations Framework Convention on Climate Change. Answer is option B. Then match the type of diseases given in list A with their causing agents present in list B. So leaf roll of potato. Leaf roll of potato is viral. So 1B. Then red rot of sugarcane, it is fungus. 2C. Citrus canker is bacterial. 3D. And root knot of tomato is a nematode. So 4A. So the answer is option B, that is 1B, 2C, 3D and 4A. Then which of the following statement is not correct for plasmid? So a plasmid cannot be used in genome sequencing projects. So that is not correct for plasmids. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.